hit all my buttons here. This is our 4th webinar in our series, and so today Dr. Rob Williams is going to talk about from candidate genes to causal variants, strategies for and examples of identifying gene variants. I wanted to give a couple of quick housekeeping updates. So what we, starting last webinar and what we'll do through this webinar is that Rob will talk for close to 45 minutes to an hour, and then we've reserved an extra 30 minutes for discussion and question and answer type session. So you can submit questions via chat, and Dr. Sen will be monitoring that chat and may save some of those questions for that discussion period at the end. There will be no webinar on July 10th. So the next scheduled webinar we're going to postpone due to some conflicting meetings for many of us in the P30 group. If you haven't received the email, we also are putting all of these presentations on YouTube. So we now have a YouTube channel too, so that you can follow and go there for when things are updated. I think that's about it that I have. As far as
anything
go.
Sorry. Go ahead, Shaunak. Are we recording yet?
Yes, we are recording. Okay,
most excellent. All right, thanks everybody. So yeah, put any questions in the chat, and Shaunak will intercept them and we'll try to answer them. I have 30 slides. I should be able to get through them with ample time. And I would like, if we do have some time left over in the first hour, to give you a live demo of some new features that might be useful for some of you. So from candidate gene to causal variants, a lot of you on the, that are on today actually have a lot of expertise on this too. You'll see this is, for better or worse, very BXD centric, because that's where we happen to have the most data at present. But I hope that gets changed over the next decade with correspondingly deep data from other labs and for the rat population in particular that we're trying to build up as part of NIDA initiatives, both what Aryl and team are doing with the P50 and what we're doing with the P30, and what Laura and team in Colorado are doing with the P20 grant that they have from NIAAA with Boris Tabakov and Paula Hoffman. So just a little outline of what I hope to accomplish today. On the right side, you can just see the link to these webinars, so you can get access to them there. And as mentioned, we've put them on YouTube. Obviously not this one yet, but it should be on there by tomorrow, along with the questions through the link you see over on the left side. On the right side are the 5 components of today's presentation. Very quickly, I just want to review some of the mapping resources and comparisons of those resources, because that's inevitably a part of the decisions that investigators have to map when they're getting ready to try to identify gene variants associated with polymorphic traits. So that's going to be quite short. There's some nice reviews on that, so you don't need a lot from me. The second part is quite a little bit more involved, and that has to do with really kind of the strategies for trying to identify gene variants. Sometimes I may use the word cloned, which is inappropriate. To clone a gene variant is sort of an old-fashioned 80s, 90s term for identifying the gene associated usually with the Mendelian variant. That term is sometimes used in QTL mapping as well. But for the most part, I'll just try to say identify a gene variant. And you'll notice that in today's presentation, there's going to be relatively modest effort on the verification in a formal way. We'll go through a couple of examples of the verification steps, but there's not going to be a lot of CRISPR-Cas9 verification, except in one case where Megan Mulligan and Greg Humanix and team we're really able to nail a variant. Third section is on classic forward genetic methods. So that's what most of us have been involved in for the last, well, many years. I will work through some examples there, but I just want to say that it is really tough to get to gene variants, the actual nucleotide variants, using classic forward genetic methods. In contrast, in the fourth topic is hybridization of reverse genetic map methods and forward genetic map methods actually makes it quite practical now to clone variants and to define genes that are polymorphic in a particular cross. And finally, time permitting, I'll give you a demo of some ways to cherry pick old findings, new sequence variants, and to do genome to phenome analysis in both directions, both forward and reverse. So that's the outline. So a couple slides on resources. And let me just move some screen items that you probably can't see that I do see. So this is from a review on resources for systems genetics, a book by Klaus Schugart and team. And this just outlines in schematic form the types of resources that everybody has, well, that most of you have been exposed to in one form or another from high school, including the inbred strains, the F1s, the F2s, the sort of Mendelian test crosses that have been used now for a long time. But there are some new additions here. Recombinant inbred strains under the number 5 are well known to most of you. Recombinant inbred intercross, number 6, RAX, perhaps a little less widely used. They've been primarily used in the collaborative cross to simply amplify the number of isogenic genomes that are available for analysis. And then under 3, there are some crosses that have been used to very effectively, in some cases, to clone gene variants. So making congenics, making consomics in particular. We had a great presentation yesterday on the use of the V6.A consomics that Joe made many years ago with Eric Lander and Singer and colleagues that are now being used to study nicotine by Jerry Stitzel and team at the IBG. The number 2 in the lower left side, you'll see RCC CICF2. I will be talking about that. That stands for reduced complexity cross and co-isogenic cross. I think the preferred term now is reduced complexity cross. And this is a unique post-genomic type of test cross or intercross or back cross that I will explain. There's a lovely paper that Cameron and Megan just published in Trends in Genetics that I'll reference briefly. And if you have any questions about that, I see Cameron was online and presumably will be online until the end. But these are some of the crosses that people always have to agonize about. What do I use given my trait of interest? How do I best get to the underlying gene variants that modulate that trait? And how do I do it within the confines of a standard R01 type support? There's a little inset there in the lower right side, which I just want to hit on this. I'll hit on it several times. There's a marked tendency, and I'm as guilty as anybody, to grab one particular resource and bang, bang, bang away at it for the rest of your career. So I could certainly be accused of banging my head against RI strains for the last 30 years, and it would be entirely justified. But it's really not the best strategy. So if somebody gave you funds for, say, 1,000 animals, and in this case, this table outlines a 960 animal study, there are many things you can do to divvy up that potential cohort into sub-cohorts that will actually give you a better return on investment, I think. For example, initially testing fully inbred strains just to understand your phenotype, make sure you can control batch effect. then testing a few F1s to see what the dominance deviations look like. Then sort of a bonus of 48, where you could either add more inbred strains or more F1s or even back crosses. Then some exploratory stages using, in this case, this was a mouse-centric article, so collaborative cross, BXD, AXB, whatever. And then using more realistic models that aren't fully inbred, but are somewhat inbred, the recombinant inbred intercrosses. So these have a much lower inbreeding coefficient than standard inbred strains, obviously. And then when you really understand your trait and you have some candidate genes, then it's entirely appropriate to improve the precision, improve the robustness and generality of the result by moving to HS stock, essentially, AI, DO, HS. Anyway, I just want to make that one point because, again, I think we do tend to silo ourselves. Perhaps it's because of the complexity of managing all of these resources, but we should consider collaborating more so that we can get better bang for our buck potentially. All of these resources have very different genetic architectures, and this figure or table rather, just quickly summarizes some of the thresholds you need in order to declare that you have something close to genome-wide significance, where obviously consomics and congenics do very well with a log that's, well, basically a p-value of 0.05 will do the trick, or less if you even know what you're looking for. And then going all the way up to outbred stock and HS towards the bottom, resources that have tremendous amount of recombinations per genome. They do require higher LOD scores to achieve significance simply because you're doing more tests per cross or per analysis. They have different genotyping costs, different costs per animal. You can build a phenome for some of these resources, you can't for others, or it's more difficult. You can study GXE obviously quite easily when you can replicate genomes across multiple environments, et cetera. So that's from the reference that's up on the upper right side, if anybody's interested in that or arguing with us. It was hard to come to a politically neutral summary, and I may not have succeeded, or Evan and I may not have succeeded, but we tried. Okay, so now on to part 2, which is strategies for getting to causal genes. And I think I have five recommendations, and at the end of this talk, it'd be great to add some recommendations because there's a lot of expertise here on the webinar, and I certainly do not have a monopoly on that expertise. It's just I really have been focused mainly on recombinant inbred strains. So there's a lot of statements about the need for high precision. And unfortunately, those statements on the need for high precision are rarely balanced by an equal discussion on the complexity of the cross and the number of variants and the number of genes in a particular QTL interval. So I wanted to introduce the notion of effective precision. So the effective precision is essentially a product of the length of the QTL. Perhaps it's 5 megabases, maybe it's 10 megabases. If you're lucky, it's 0.5 megabases. And the number of variants that are segregating in your particular cross, which you do have some control over globally. You won't have any great control over it for a specific locus, but at least in general, you have control over it. So for example, the HS rats will generally have 3 to 5 million variants segregating from the 8 genomes that went into that cross, maybe more. Abe can correct us, or somebody or Hao Chen can. The BXDs have about 6 million segregating variants, but in a very patchy format. But it's not just the number of variants, it's their patchiness, and it's the patchiness with respect to the local density of genes. So we really need to start thinking about the effective precision rather than just the absolute thumping our chests about our 0.1 or 0.2 megabase precision. It doesn't do you much good to have 0.5 megabase precision if you still have to sort through hundreds of candidate genes and variants. And just one cautionary example on the bottom of this slide. So this is the first QTL I ever mapped in 1998. It's a gene that controls the number of neurons in the retina that project into the brain, so-called retinal ganglion cells. The LMM output from GEMMA is in the upper right. Nice QTL on chromosome 11 with a lot of about 5. This is zooming into that region. I don't know if you can see the little blue dots right there are markers with LODs of about 5, a little bit less, I think 4.68. The interval is, by my standards, mapping with RI strains, impressively small, so in the neighborhood of 2 megabases. You'll notice that's not formally true, so the 1/2, you could go over here if you use the 1.5 LOD drop-off interval to define the QTL. But because the SNP density is so much greater between 98 and I think that's 90, this is 99, this is 99.5. So we basically have a 1.5 to 1.7 megabase interval with high SNP density segregating between the B and D parents. It's our best candidate. But, and here's the killer, there are so many genes. This is essentially a Manhattan of the mouse genome. It's one of the Hox keratin complex clusters. There are 4 of these in most mammals. They're just famously gene dense regions. And if you map a QTL into one of these regions, the response should be, ouch, you've got a lot of work ahead. I've studied knockouts in 2 of these genes. I have a candidate, but it's been 24 years now, and I'm really not very confident about that candidate. So effective precision has to take into account these not so secondary variables, gene density and SNP density. Strategy 2, and I'll actually talk about this in the context of the previous example. We now have the luxury of sequence data, and it's surprising how ineffectively most of us use our sequence data, myself included. We've made an effort to use it, but I find that I don't use it as effectively as I should. In this case, I'm showing on the right side a variant browser. It will display either SNPs or indels, and now that we, thanks to David Ashbrook and a team, a large team of BXD sequencers, and Hao Chen and team for the HRDP, we have full genome sequence and longer variants, not just short indels and SNPs. We have inversions and structural variants and STRs for these strains. Those need to go into a variant browser. And in this case, what you're looking at is I typed in, actually, I didn't type in chromosome 19, I typed in chromosome 11, and I put 98 to 99 megabases. I then trimmed it down to just Black 6 and D2, the parents of my cross. And then I said, please show me all of the non-synonymous variants. So here you see all the protein coding variants in that interval, including 2 protein coding variants in cancer susceptibility candidate 3. NR1D1 is actually the thyroid hormone alpha receptor, which is my personal favorite candidate. And it does the knockout does have a phenotype that's about what I expected given the phenotype segregating the BXDs. But it's basically too much work. I would probably have to spend close to $1 million to actually validate which gene it is. And frankly, I'd rather be doing things that scale better than just studying one gene variant that affects retinal ganglion cells. So in 1999, when we had this problem, we decided, okay, to get this to scale, we have to build resources that make this possible, not just for one particular QTL, but across the whole genome. And that's, well, that's been a major motivation for my work. So getting the DNA variants is certainly much more feasible now. The work that we've done with the BXDs has involved linked reads. We have parental data using Oxford Nanopore. So we should be able to get a fairly comprehensive list of all variants except in the ugliest parts of the genome, such as the Y chromosome and telomeric, centromeric regions. Strategy 3, whatever that says. I can't actually read what it says. I think it says make sure you can get a phenome. So the more data you have, the more likely you'll be able to make progress in going from candidate genes, which we talked about 4 weeks ago, to actual genes that are sufficiently well filtered that you're motivated to consider secondary attacks to validate and verify that you've got the right gene. So a deep phenome works. You can do this with either an isogenic cohort. You can do it with consomics, BXDs, HRDP. You can also do it with an HS or the DO, but you really have to have your act together. You have to build up a large collaboration so that you actually generate a lot of data for a particular cohort. The power of replicability, sorry about the typo, can be a major win. And that is something that's harder to overcome with HS or DO, but it's pretty straightforward to do that with any kind of isogenic cohort. So in the case, Shuhita Roy and I have been working on longevity in the BXDs. We have longevity data for 70 strains with 10 replicates. And I can tell you that those data for merely 70 strains outperform the Het3 four-way F2 intercross, for which we have 6,000 cases by a wide margin in terms of precision of loci. So you'd think that an F2 with 6,000 individuals would have good precision, but our smallest 1.5 lot interval there is about 10 megabases, even with 6,000. Whereas with the BXDs, we're able to really get on top of a few candidate genes for longevity with intervals, confidence intervals, support intervals of under 2 megabases in a few cases. So, and then we can combine that theme of this particular slide with omics data we have for the same strains of mice, for the same ages, for the same treatments, for animals with known longevity or known longevity distributions, I should say. This I already hit on once, and I promise to say it again. Don't just use one resource to try to, quote, clone a gene variant that's causal to a trait. Use as much as you can. The classic, and I think in some ways the best and most translationally relevant method, is to hook up with somebody doing GWAS studies on a related trait. And if you're that's not always possible, but if your trait is something that's quite where there's no argument about the homology of the phenotype, blood pressure in mouse and blood pressure in human, nobody's going to give you grief in study section about whether you're measuring the same thing. Well, they might because of study section, but it would be unjustified. We've gotten a lot of flack now from Josh Gordon and NIMH about, what does psychiatric disease mean in a rodent model? I find that frankly offensive, given all the progress that rodent research has contributed. Just look at Tom Insel's work, if you need an example, the previous director of NIMH. So even for psychiatric diseases, there's a lot of progress you can make. And but in the case of blood pressure or Aryl hydrocarbon receptor, this is a study, both of these studies actually in collaboration with Johan Owerks. One used human GWAS from a Finnish cohort to resolve which of 3 particular variants mapped in the BXDs was the strongest candidate. And this upstream binding protein 1 was the strongest candidate in the finished cohort, not by a large margin, so I don't consider this definitive, but the biology of UBP is what really makes it definitive. And if you have a look at that paper, there's a mechanistic interpretation of the finding and how UBP, testosterone, etc. So a pretty compelling candidate. And again, it's not necessarily worth $1 million to make the CRISPR-Cas9 and do the detailed phenotyping. It's really more important to move on to possible studies in humans at that point, or other GWASs. This was an old study. This was done in, I think, 2010 or 8. So there will be large GWASs that would be appropriate to extend this at this point rather than working in mouse. Or one could actually revisit this in rat now that we have stronger rat resources. Aryl hydrocarbon receptor, this work was confirmed in humanized mouse, Drosophila melanogaster and C. elegans. In all of those variant models, aryl hydrocarbon receptor had an odd effect on activity level, which I don't think anybody expected. Obviously, it's an important transcription factor, but to the best of my knowledge, until this paper came out, nobody had said that it had anything to do with activity level. And really, since this was published, nobody has bothered to follow up on the neurobiology of how AHR is actually modulating activity. Well worth doing. Molecular endophenotypes. So if your heart is set on a particular research problem, or I should say a particular range of phenotypes, say drug abuse, response to nicotine or cocaine, then what I'm advising here may or may not apply. But it's much is maybe the too strong a word. It is easier to work with molecular endophenotypes rather than your higher order trait. So if you're interested in substance abuse, which many of us on this webinar are, then it's actually easier to deal with molecular endophenotypes associated with substance abuse. And we obviously have 40 years of beautiful molecular biology on substance abuse. So we know dopamine obviously plays a great role. So if you go into Gene Network, you can look, or other resources, you can go in and look for endophenotypes related to dopamine. I don't know if you can read that in the lower left side, but it says central nervous system pharmacology, protein expression, dopamine receptor 2 and 3 binding maximum, so the Bmax, in membrane fragments from the dorsal striatum. So work that Byron Jones and team did in 1999 with when Jerry McLaren and Bob Ploman and Cheryl Reed. And so it's not a very deep study. So data for, I think, 17 strains in there, but it's a nice molecular endophenotype. It'd be relatively easy to extend it to 100 BXG strains would be practical right now. The one at the bottom that's highlighted is a study from Crabb et al. I think the last author in this case was Belknap, Aaron Jakowski, and Tamara Phillips and Chris Cunningham, sort of a who's who of the Oregon group. And that trait is central nervous system pharmacology, protein expression, dopamine transporter. Old data, both of these now 20 years old. And I'll show you what you can do with them, because it's amazing what you can do with these old data sets now. On the right side, you'll see Johan's team's approach to system genetics and metabolism. The advantage there is that many of the core phenotypes that you're interested in to begin with are essentially endophenotypes. So here are a lot of blood chemistry traits. And if you look at the column, I think third from the right margin, that's labeled maximum LRS, those values are all very high, 32, 31, 31, 31, 30 for traits such as mean red volume, red blood cells. And you notice that red blood cells, chromosome 7, 104 megabases, you can almost clone that one in your head because that is very close to the position of HBB, hemoglobin beta complex. A lot of these are easy to clone. So you'll see a list later on of traits that have been, where the likely gene has been identified. And in general, I don't include these that are just so easy, but a lot of these are worth going back to. And what I will show you is going back to the two that are highlighted in yellow on the lower left side. So, the heading here says, does 5 work? Does strategy 5 work? So I'm going to take you through 2 quick examples of how this might work. This is the Jones paper. On the kind of to the right side, you'll see the GEMMA Manhattan plot with a very high minus log p-value on chromosome 15. This is pure chutzpah because there's 17 strains in this data set. So I just want a big caveat here. So, but you'll see that it actually makes a lot of sense in the end. Although here, the other cautionary note is if you're motivated, you're always going to be able to narrate a story around your personal fantasy. And I'm going to be guilty as charged and actually narrate around this particular QTL. I like the lot of nearly 8, but it really needs to be replicated with at least another 2 or 3x increase in numbers of strains. But it would be entirely practical. This is a summer project. Just get a bunch of striata from our biobank and do your thing with the Betamax. Right under the Manhattan plot is a Manhattan plot zoomed on chromosome 15, just showing that it is a gene-rich region, which is a bit annoying. But there's a wonderful candidate gene in this region, ataxin 10. And I knew nothing about this until about 2 a.m. this morning. But ataxin 1 has been associated with downregulation of dopamine receptor. This is in cerebellum. But I think in conclusion, we can say that we have a good candidate here. Strategy number 5 does work. The end of phenotype is pretty easy. Obviously, we haven't cloned or identified anything, but we have gotten to a point where we have a good hypothesis that's worth validating, and validating it would not be a major motion picture. A second example. So this was the other trait. Also an embarrassingly low number, but not quite as low. I think 23 in this. LOD score, also pretty damn good. I think about 5.5. The gene that they're actually mapping as a trait, so this is really a molecular trans-eQTL. So the DAT gene is located on chromosome 13. The QTL is actually on proximal chromosome 19. It's got the enviable situation of mapping into a region with very low gene density. So you'll see that in the zoom region, right? I think you can see my cursor going around in circles. So we have a fairly small interval of about 1 to 1.5 megabases. There are almost no genes in here. So again, it's gene sparse. So this is a more straightforward candidate. And in combination with sequence data and CRISPR-Cas9, you could probably within a year, and probably without blowing $1 million, define a very interesting trans-modulator controller of DAT expression, which would be big. That could be quite a coup. And the obvious candidate in this region, so this is maybe 3 a.m. today, is the PSAT1 gene. It's really the only strong candidate in this region that I could identify. There might be something bizarre in there that's escaped my attention, but this is a gene that, at least in a case study, I mean, this is very sparse data, has been associated with some variant of schizophrenia. So it's got some plausibility here. Okay. So that is pretty much, I'll say a little bit more about identifying genes, but I want to get to the topic of really nailing a gene. In other words, making everybody happy. The grouchiest reviewer on study section will acknowledge that you really nailed it, that you're not just using your ability to spin a yarn, the narrative potential, that you really have a locus. And I would say that in general, this is impractical. I hate to say it because it has been my goal, but after having battered my head against this goal for 20 years, I acknowledge that there's smarter ways to get to the variant, but there's a huge caveat there. Do you actually need to know the nucleotide variant? And I would argue that most of the time the answer is no, you don't actually need that nucleotide variant. It's not going to do you much good. And we can talk about that and argue about that later. But here's an example of a case where I think it'll be obvious to you why trying to get to the nucleotide is pointless in this particular case. So this is some data from Alyssa Chesser's group when she was at Oak Ridge National Laboratory. Vivek Phillips, first author on a beautiful paper. It should have been in Nature Genetics, in my opinion. Just a ton of omics data related to substance abuse. It was a big NIDA-funded R01. And this is looking at the response to morphine, just the activity level. A huge QTL on proximal chromosome 10 with a log of about minus log P of about 10. The locus is relatively large. So you'll look at that, what I just put in there. It's what? It goes from about 3.5 megabases to 9.5 or even 10 megabases. But there is just a brain dead obvious candidate gene here, and that's the mu-opioid receptor. So at that point, you know, different philosophies for different people, but at that point, you do need to review all of those genes. So Boris Tabakov at one point definitely thought there was another candidate in this interval, and I should probably go back to his paper on that. But for me, this mu-opioid receptor is the no-brainer. And but you can see, and I'll show you some other evidence that it probably is the gene. That is a huge interval. It's highly polymorphic, and it's even worse than that because it turns out the mu-opioid receptor is itself full of many sequence variants that modulate expression. So this is where the omics data comes in handy. So here's omics data, some from Mike Miles for the ventral tegmental area that shows a high D allele, some for striatum, for hippocampus, and for adrenal that shows a high B allele. So an additive effects over here tell you who's got the high allele. Negative means the B allele is high, positive means the D allele is high. All of these cis EQTLs are ridiculously strong. They're very close to the gene. And the bad news here is finding the causal nucleotide variant associated with these cis EQTLs is just impractical. It's just, it's not worth the work. And the reason is that there are probably on the order of 2,000 variants in here. There's probably not a single causal variant working on the expression levels of mu-opioid. They're probably splice variants or all kinds of things that can happen. So this just shows you in this case, we can say something. We can say there are no protein coding variants in this gene segregating the BXDs. So that's what I'm showing you here. So we can look at the OPRM1 cis-EQTL in the upper left. It's a whopper. That's a minus log P of 20 some. It's a monster gene. Well, not monster, but it's 280 megabases in extent. So a lot of variants there could impact expression. no missense mutations based upon our variant browser. There are some variants, obviously, but they're intronic and nothing too exciting. So it'd be a major effort to try to understand what's controlling the expression, and you really have to ask yourself, why? Why do I need to know that? And I would argue that in general, it's not necessary. I'm going a little bit too slowly. This is a lovely analysis of when it is possible to get to the variants. So in this case, we have another one of Byron Jones's, actually this is the same trait I showed you before. Or is it the same one? Yes, this is the Bmax in the striatum. It maps into this region of chromosome 16. It maps into a region that's identical by descent. It's a fairly large region, so it goes from 15 to almost 25 megabases, but I can tell you with considerable assurance that we know what that variant is. So this is an extreme serendipity situation. We've mapped a trait related to dopamine, a monoaminergic trait, into an interval that happens to contain the catechol O-methyltransferase gene, which is right where that red arrow is. Actually, it's a little bit off from where that arrow is. So here is the catechol O-methyltransferase trait in that IBD region. So this is a region that's identical by descent, and yet we have this huge cis-QTL here. And the question is, how did we get a cis-QTL in the middle of an IBD region? And the answer is this B2 sign element got inserted into the 3' UTR of BLAC6 and produces a premature polyadenylation site. Actually, it produces three premature polyadenylation sites that change the structure of the 3' UTR quite dramatically from a long form in D2 to a short form in BLAC6. And a lot of subtle phenotypes are downstream of that variant. And so at this point, we're beginning to enter into reverse genetics because we now have a sequence variant, a B2 sign, polymorphism segregating the BXDs, and now we're asking a reverse genetic question. What consequences does that sequence variant have on gene expression in nucleus accumbens, in hippocampus, in prefrontal cortex, in striatum? And these are all transcripts that are modulated by this IBD region, almost certainly caused by the short and long form of 3' UTR and some other differences probably. And then we can go out a level and get to Byron Jones's trait, which was DRD2 density and beta max, and in his case, the caudate putamen. But it also modulates DRD1. It modulates some traits that Bob Hitzman and team studied. So these are drug response traits. So we, for those of you who want to look at this, this is the reference down here. Megan Mulligan and Lee were the lead authors on that. And it's probably our best or first example of using the BXDs as in a segregating population for reverse genetics. Then in 2016, we had a paper with a lot of people, Johan and Josh Denny. And this is I'm going to whiz through this, but just to say that in this, we're doing phenome-wide association in using the BXDs, which has all kinds of problems that are mentioned up in the upper left corner there. The linkage disequilibrium, the LD, is the main problem for using or doing phenome-wide association in a mouse, or at least in the BXDs, because the linkage blocks are very large. But that's why we teamed up with Josh in the BioView group at Vanderbilt to do this jointly, because we get precision from the humans, but we get quantitative trait from the mouse. And here you can see the phenome-wide association in the mouse for a particular trait. I think this is one of the alkaline phosphatases. Oh no, this is for collagen 6A5, modulating bone mineral density, which is sort of a no-brainer. And then this is the corresponding PHWAS from BioView for this particular gene, or allelic variants in that gene, and some traits from the BXDs and the parental difference. So we did this a couple of ways. This is the AHR that I already introduced you to, just showing the expression difference and some of the traits associated with that, with the mouse phenome-wide association over here on the left and the human PheWAS on the right. So this is a potentially powerful method, but you really have to have both mouse and human to get it to work, at least for our group, because of the LD problem. So getting on to the fourth part. So using the sequence data, I mentioned that as important. Rather than show you, well, maybe I will. Let me just show you what this should look like. So this is the supplement to the Wang paper, this joint PheWAS in mouse and human. It's mainly BXD. Here are all the tables, and I'm going to show you this table for all of the missense variants segregating the BXD family. Easy to download this data set, but here they are. There are 12,000 of these. So you know, most of you will not be interested in olfactory receptors, but you can ask a question. Do I have anything in a GABA receptor? And the answer is no. But this is really a kind of sea change in how to do reverse genetics. Reverse genetics typically is involved in mutagenesis or knock-in methods, and has not been on a segregating background. But now you can take the relatively small effect alleles that are generated by missense variants in a cross such as the BXDs or the HRDP or the HXB rats or HS. Doesn't have to be an inbred panel, just have to have good sequence data. And you can say what phenotypes are downstream of the variants here in RP1. And it's surprising how few people are taking advantage of this method that essentially has been kicking around now since we first got mouse and rat sequence data. So it's now close to 15 years. And I would say, to the best of my knowledge, nobody's made a business out of doing this. Abe is getting pretty close and is really doing a good job getting all the sequence variants out of mouse populations. We're trying to do the same for the BXDs, and Howe is doing the same for rats. So we should be able to do this very systematically, provided we have a phenome, which is the kicker here, getting the phenome. So I prepared this slide about 5 years ago after I got sick of hearing study section members say that you can't clone genes or identify genes in the BXDs. I haven't updated it for the past 3 years, and I should, but even 3 years ago, we had at least 20 genes where I feel quite confident that we have the causal gene underlying the phenotype. So I've highlighted COMT in red. I've just talked about several of these. I will take you through GABA alpha 2, AHR I've mentioned. Tamara Phillips has a fabulous finding of a variant just in the newest model of the BXDs that modulates methamphetamine addiction. A series of papers there with Aaron again and Tamara. And that's been a real eye-opener. But for those of you who need boilerplate to defend your approaches, that's what that was, why I did that. Oops, let me get back to this mode of epoch effect. I think I'm going to have to skip over epoch effect more quickly than I intended, but the BXDs are quite unusual in that people like Lulu and myself have been extending them, and Ben Taylor extended them quite a bit. So the original Taylor series of BXDs were generated starting in 1970. They're now up to F125 on the breeding. Then Ben's made about 10 new BXDs in the 90s. We started making them using advanced intercross strains in the late 90s. So that's this third epoch. The fourth epoch was just a conventional F2-derived set of BXDs that are not quite as recombinant. And then we got some advanced recombinant inbreds from Abe at Chicago when he was doing an advanced intercross of B and D. We made some ourselves. So these are a second set of AI-derived BXDs. And then there's some strains here that we're making now. We haven't finished making them, so we don't really have a good tally of the total number of crossovers per animal, but we expect it to be, it should, for a conventional F2-derived RI, it should be about 40 recombinations. For these AIs, it can be at least twice that. The epoch effects turn out to be incredibly useful. So David has a paper that we've just submitted. It's in bioRxiv if you want to look at a fairly mature version of this paper. And I will show you how epoch effects, why they, as Alyssa would say, why they rock. So this is our first exposure to an epoch effect, and it's the only case where we really know exactly what's going on to the nucleotide and have been able to repair it. I mentioned this is Megan's work. So the GABA alpha 2 expression level in the hippocampus is not segregating at all in the first set of BXDs made by Ben Taylor up to BXD 32. But then in Taylor's second set and all subsequent sets of BXDs, expression level of GABA alpha 2 does segregate. And so the first question is, what the heck is that all about? And the answer, quite simply, is that there was a new mutation that impacted C57 Black 6, our classic wild type strain, after 1990. We know that for other reasons, but so we have pretty good time on when that mutation happened. That was before JAX was repeatedly rederiving from cryopreserved stock. And then when these BXDs, when the matings were set up in 1990 by Ben, the BXD, I'm sorry, the Black 6 female that he used had that mutation. And so all of the progeny, or roughly half the progeny, had that mutation. And now this becomes, it's tempting to say a no-brainer to identify the nucleotide, but it did take us about 7 years. So here is the CRISPR-Cas9 design by Greg at the upper part of the figure. Megan did a great job sequencing this every which way using whole genome sequence, but then doing a lot of Sanger resequencing. The SNP, well, I should say the indel that really is operating is right here, right at the start of exon 5. So this is reading from your right to left. And there's a variant right here, right next to the splice acceptor that is the culprit. And we know that because we fixed it and all of the problems went away. So if anybody wants a real Black 6 circa 1980, we have it. And now JAX has it. Not that anybody cares at this point. They're all perfectly happy to work on their GABA alpha 2 mutant. All of the comped mice are in a Black 6 NJ. They do not have this mutation. So this is restricted just to the classic JAX version. But then you can now use this GABA alpha 2 known mutant and the repaired version from Greg and team and Megan, and you can ask simple questions about what is downstream of this variant simply by comparing, classic knockout versus wild type. And this just shows some of the molecular downstream consequences on other GABA alpha isoforms, or I should say proteins, and mRNAs in this case. How do they respond to the fact that the expression level in Black 6 is half of what it should be? And by the way, the protein level is roughly half of what it should be. So we can now do reverse genetics. And I should say this sort of reverse genetics actually traces back to some work that Vivek did with Joe Takahashi on Sci-FIP2, probably the highest profile work of this type. We also did this many moons ago in looking at heterotopia in VXD29, what we called a co-isogenic cross. So this is now a damn powerful way to combine a segregating population with known variants, which brings us to this reduced complexity cross. So I'm now just about at 1 hour, and you can see I'm at slide 26 of 30. So I'm just going to speed through this and just say that there's a great paper review that just came out from Cameron and team, Megan, in Trends in Genetics. If you want to know more about reduced complexity crosses, they have a nice table that shows all the reduced complexity crosses one could and should probably make. There's a BALB-C family that could be done, an AJ family, a C3H, and obviously a lot of black 6s and 129s, et cetera. And the same obviously can be done in rat. So we could redo this reduced complexity in rat. And in fact, Megan and Hao Chen and Eva Radai just got funded to actually do a Wistar Kyoto reduced complexity cross to track down a known phenotype variant that affects all kinds of depression-related and behavioral outcomes. So we'll have a great example in rat in short order. Okay, so some conclusions here. So number one is don't obsess about precision. Use as many resources as you can. Number two is don't obsess about getting down to the nucleotide. Usually you don't actually care. If you're serious about mutation spectrum, if you're a hardcore geneticist, then sure, go ahead and obsess about the type of mutation and how it operates on the phenotype. But I'd say most of us are actually interested in translation and genetic mechanisms, molecular mechanisms, and not in noodling around with mutation types per se. I would say by far the most effective way to identify genes and identify variants, nucleotide variants, is now to use these hybrid methods where you do a little bit in the forward direction and as much as you can in the reverse direction, using as much omics data as you can get your hands on. And I just want to close by saying, take advantage of earlier studies. There's a ton of great data that is just sitting there waiting for a grad student, postdoc, or even a senior faculty member to clone it, quote unquote. And I was going to give you a live demo of this, but that's going to have to wait. But believe me, it's amazingly easy. I showed you two examples from the Jones paper where I got a good candidate and the Jankowski paper where I got a good candidate, PSAT1. I think both of those are worth follow-up because the added phenotyping for these particular endophenotypes is pretty easy. And this I will skip using outliers, but I can say that David and Hao and others are generating wonderful lists of mutations that are specific to single strains of mouse and rat, and that's a gold mine also, because those phenotype outliers can now be combined with the genetic outliers and with reduced complexity crosses to nail them probably if you did this efficiently within 2 years for any one of these. Okay, so there I am. It is 1:03 or 4, and I'm done and ready for input, and I'm gonna stop my screen share.
So, Rob, the first thing, I think there was a comment by Arthur that the slides and the PDF material are on opar.io.
Oh, great, terrific.
Of course. The second question or comment was from Joe Nadeau. I think, Joe, are you still there? I am not. Okay.
I'm looking for him.
He's not there, I think.
Yeah.
But can you see his comment? So he says that by using lots of different strains and resources, is there an implicit model that the QTL has a pretty constant and consistent effect across strains and background? That is, not much epistasis, background, or context-dependent effects. Does strong independent effects limit the scope of discovery? QTLs whose actions across backgrounds, of course, are pretty interesting, but the others are interesting too. It's probably important to understand the strengths and limits of the underlying model that's being tested.
Yeah, so this is a great point. And I was thinking about putting in a caveats to this entire presentation. So this entire presentation pretty much assumes that you're chasing after an additive effect locus, or maybe 2 or 3 additive effect loci, and you're not, but as soon as you actually do what I suggest, get involved with multiple crosses, then you definitely have to consider what Joe wrote there, which is, and you do with the diversity outbred, with the collaborative cross, with the HS, you have to look at the actual haplotypes that are generating most of the variants, and that gets a bit tricky. Abraham Howe, a lot of people have more expertise than I do at this, Jonathan Flint. So yeah, you definitely need to have a more sophisticated model that basically that looks at the haplotype contrast crudely in all different pairwise combinations. The very first study that I mentioned, looking at retinal ganglion cell number, we actually did the BXDs and the BXHs, and there's some dramatic differences there that we had to take into account. We just ran roughshod over those differences, which you can in some cases, if you know the same allele is segregating in both crosses. But if you don't think, they're completely different resource types, then everything Joe said is absolutely right. And epistasis and G by E and robustness all really have to be taken pretty seriously. So if you do your study in sequence, you do all of the BXDs one day and all of the DO the next day, you're going to have a potential for a batch effect and GXD effects and drift, etc. So it is technically hard to do that, what I recommended.
So the next comment was from Cameron Bryant, who had to leave because he had another commitment, just as you were talking about reduced complexity crosses.
I love that comment.
To the, I guess, the junior investigators, don't tell people at your chalk talk that you won't need the nucleotide variant. You will not get the job. What do you say to them?
That's a great comment. But I would say, when you get to be 68 years old, you can push back and tell them to screw themselves. You really have to say, what is the justification for having that nucleotide variant? In the case of GABA alpha 2, where we do have that variant, what does it buy us in terms of translational utility? We already knew that the GABA alpha 2 effect was working in expression domain. It was not a protein variant. So we had everything we needed to do anything downstream, just knowing it was a cis-y QTL, and actually repairing the nucleotide was great. I'm delighted that we did it. But really why that GABA alpha 2 animal model is so cool is because what we can do to get at downstream phenotypes, just as if it were a knockout. It's a more subtle knockout that only changes expression and possibly isoform use. So it's really I think most people at Chalk Talks should appreciate that the goal is not necessarily a nucleotide, although they may not, if they're heavy-duty molecular biologists, but the goal is really to understand the sources of phenotypic variation in human usually. And your nucleotide variant at the splice acceptor site of exon intron 4 is not going to generalize to humanity. It probably won't even, frankly, it won't even generalize to another strain of mouse or cross, unless it's got the new model C57 black 6 in it. Okay, yes, I saw that one, Shaunak.
Well, I think at this point, others can also chime in, unmute yourself and ask a question or raise your hand, whatever works. As people think about it, let me ask one question. This is maybe from the more quantitatively minded people, which is, what can you define endophenotypes for us?
Oh, it's a squishy word, but it means, obviously from the etymology, it means sort of an internal phenotype. So it is opposed to most of the phenotypes that, well, I guess even in a clinical setting, there are two types of phenotypes one deals with. One deals with typically dichotomous, has disease, does not have disease. It's sort of the case control categorization that dominates much of clinical work, or an ICD-10 or code that is critical for billing and now becoming, unfortunately, more critical for phenome-wide association. So those we consider, so I guess anything that is a higher order phenotype, a behavioral pattern, type 2 diabetes, even if you have a quantitative measure, type 2 diabetes would be considered a proper phenotype. And I put big quotes around proper. An endophenotype, on the other hand, would be something that requires often a molecular assay. Frankly, all blood chemistry could be considered endophenotypes. Any metabolite, metabolism, if you went into humans or mice and you counted the number of beta cells in the pancreas, I would consider that a cellular endophenotype. But you can see where one person's endophenotype is another person's core phenotype. So if you've spent your entire life studying the genetic control of RNA expression, you might consider RNA to be your major higher order phenotype, whereas most of us would consider an mRNA expression estimate to be a molecular endophenotype.
Is this what some people would call a mediator? That is not, it's not the phenotype itself, but is something that causes the phenotype?
Oh, perfect question. Let me share my screen. Thanks for reminding me. Okay, let's see. Oh, people can't, I don't mean to share my screen. I mean to share myself. Stop screen share. Can people see this? I think, Shauna, can you?
Yes, we can.
Okay, so the concept of mediator is really critical, and I hope there's quite a bit of interest on our side of these webinars to actually give a series on more complicated models of the relationships between genome and phenome. And you can do this using structural equation modeling or Bayesian networks. And what Shaunak just asked about mediation, in other words, if you go from a SNP in GABA alpha 2, you know, because of what I told you, that it's not mediated by a protein variant because there is no protein variant, but you know it is mediated by an expression difference in alpha 2. It could be a slight difference in isoform usage or transport of mRNA, but ultimately it's expression difference of the mRNA. So that is the first step. And since it's a strong cis-QTL, there's really nothing upstream of that mRNA cis-QTL other than that nucleotide deletion in black 6, or I should say the contrast between black 6 and D2. So we can then define that as a mediator. So we go from SNP to alpha 2, and then all hell breaks loose in a molecular domain. And Megan, in that figure from the Frontiers paper, showed you that first level. What are the consequences on other GABA-A mRNAs? How do they respond in different chunks of the brain? Who goes up and who goes down in response to the change in alpha 2? But then there's going to be a whole cascade that eventually leads, and this is Megan's ongoing work, to changes in behavior and response to alcohol and other substances. So mediation is now becoming more practical. The software is there. Often the sample size is not there. So what I showed you today, the sample sizes are embarrassingly small, and you really want to be, well, you know, hundreds, 200s, 300s would be kind of the beginning comfort zone for most of these more complicated models where you have 5 or 10 potential mediators that you're looking at. But Shaunak, you answer that question because you know actually more than I do about this.
You know, we were hoping to discuss this at some point in the future, right?
Yeah, if anybody's still on, so the 35 of you still on, wants to know more about structural equation modeling and genetics or Bayesian network or simple stuff like just mediation analysis or partial correlation, that would be, we'd be glad to put something together. There aren't that many worked examples of this yet, but we're getting there. Lulu and I just did a fun one for ACE2 expression in an influenza model that we hope has something to do with COVID-19. We'll find out.
So everyone, please chime in with questions. You can write on the chat, unmute yourself and ask directly